Morning everybody. I am here to teach you the topic protein synthesis or translation. Protein synthesis means synthesis of new protein from the DNA via the RNA. This process is also called as translation. So translation is basically the process of synthesis of proteins from the new protein from the DNA via the mRNA. So here synthesis of RNA molecule from the DNA, that process is called as transcription, and the enzyme responsible is called as RNA polymerase. Then there are three different type of RNA: mRNA, tRNA and rRNA. So mRNA basically involves in the process of synthesis of protein, so translation. So this mRNA is translated into the protein with the help of several enzymes, and the machinery responsible is the ribosome. So central dogma of molecular biology tells that there will be the unidirectional flow of information from DNA to protein via the RNA, and synthesis of new protein stands for the growth and development of the new trait. So this is called as Teminism also, because the two scientists Temin and Baltimore devised the process. So here, since we are telling that there is synthesis of protein, so protein synthesizing machinery over here is the ribosome, which is present in the cell cytoplasm. So this protein synthesizing machinery requires different raw materials like any other factories which requires raw material to synthesize its product. So here for protein synthesis, raw material is all kinds of RNAs like mRNA, tRNA, rRNA. Then DNA, obviously it will be required for specificity control. Which protein has to be synthesized, that information lies within the DNA only. So DNA is required for specific protein to synthesize. Then the building block, that is amino acids, which is present in cell cytoplasm as amino acid pool. Then there are different need of different enzymes, then energy precursor molecules ATP, ZTP, and then enzyme catalyzers like Mg, magnesium ion is also required. There are different RNAs with distinct function are devised in case of protein synthesis. So three different type of RNA: mRNA, tRNA, rRNA. mRNA is present synthesized in nucleus, goes to cytoplasm for synthesis of new protein. tRNA, it is also present in cytoplasm. It provides linkage between mRNA and amino acids, transfer amino acids to ribosome. Then rRNA, which is present in cytoplasm and which is structural component of ribosomes. mRNA, it is the messenger to transfer the message from DNA to the protein. Then the machinery ribosome. In case of eukaryote, it has two subunits, 40S and 60S. 40S is a small subunit which has a stretch of 18S RNA and 33 different protein, whereas the last subunit has 3 stretch of RNA, 28S, 5.8S and 5S, and different 49 different proteins are also required. So together ribosome is 80S. Then we can see the internal component of ribosome. It has three sites: EPA. E stands for exit site, P stands for peptidyl tRNA binding site, and A stands for amino acyl tRNA binding sites. There are two different type of ribosome: free and membrane bound ribosome. free ribosomes are present in cytosols, and the protein which is synthesized on the free ribosomes remain in the cytosol, and that may be act as structural or enzymatic part of that cell protein of that particular cell, whereas those ribosome which is bound on the surface of rough endoplasmic reticulum synthesize secretory proteins, and targeting and sorting of that protein only occurs. tRNA, as it is a small extremely stable RNA structure present in both prokaryotes and eukaryotes, here is the structure of tRNA, where 3' end is for binding of amino acid, whereas the D loop near to that there is the loop called anticodon, where this tRNA binds with the mRNA. This is the shape of tRNA. Then there are 3 steps in protein synthesis translation: initiation. Then in initiation, the 2 subunits of the ribosome come together and start codon on the mRNA in the rhizome to set the reading frame, and there will be the synthesis of first peptide bond. Then elongation, charged RNA attach and peptide bond from between the amino acid, and ultimately the whenever there will be the any termination codon comes, then there will be the termination of protein synthesis. So mRNA translation mechanism has 3 steps. Step first stands for an amino-acyl tRNA. This amino-acyl tRNA is also called as charged tRNA, binds to the A site of the ribosome, and that A ribosome, that site also binds with the mRNA. Then there will be the binding of large subunit. Then this formation of this initiation complex is called as formation of translation initiation complex. So next what happens, a new charged tRNA is that tRNA which binds with the another amino acid molecules that comes and binds at the site A, and then there will be the formation of new peptide bond. So elongation stands for formation of first peptide bond till the addition of last amino acids, and then last process is called as termination. So whenever there will be the any termination codon comes in the process, so there will be the complete termination or stoppage of the protein synthesis, so that is called as termination. So whole process is called as translation.